For the second session of Covering Rare Diseases, Day 3, we're joined by Dr. Anna Greka. Anna is a professor of medicine at Harvard Medical School in Cambridge, Massachusetts, a physician at Brigham and Women's Hospital in Boston, and she is an institute member of the Broad Institute of the Massachusetts Institute of Technology and Harvard. Now, you can read more about Anna's long and distinguished career and accomplishments on our website at nationalpress.org, but I'm going to take a stab at one sentence. Anna leads a program aimed at dissecting fundamental mechanisms of cellular homeostasis and their disruption in genetically defined kidney, metabolic, and degenerative diseases, with a special focus on membrane proteins. Now, that's a fancy description, and I'm surprised I made it through without tripping over my tongue. But through her presentation, Anna Greka will help journalists understand how her research aims to break the chains of inherited disease for many rare disease patients. Anna Greka, thank you for joining us today.
Thank you, Rachel, for the very generous introduction, and it's my great pleasure to be with you today. I am grateful for the opportunity, and also I imagine that many of you in the audience are in different time zones, joining us from, different parts of the world. So it's a special honor and pleasure to be with you today. So I will try to describe some of our recent work in the area of rare genetic diseases. And tell you a little bit about a new initiative which we're hoping to advance over the next few years to really try to make progress against as many rare genetic diseases as possible. So I'm just making sure that I'm loading up my slides. I will be happy to take questions at the end, but also if it's convenient to interrupt me in the middle, I really don't mind. So that's up to all of you. So I want to tell you about a new initiative I'm building called Ladders to Cures. As Rachel mentioned in the introduction, I'm an institute member at the Broad Institute of MIT and Harvard. This is an institute that's very well known for our role in the Human Genome Project. So basically understanding the 3 billion letters of genetic information that make up our DNA, and then trying to decipher what it means in the context of disease. And the most straightforward area of human disease where the Human Genome Project has had a huge impact is actually in rare genetic diseases. So as you have probably been hearing over the last few days, rare genetic diseases are caused by mutations in a single gene, a defect or a misspelling in a single gene. They are collectively common. They affect more than 10% of the population, as I think you've also probably heard. There are an estimated, and we can argue about this, but there are an estimated 8,000 genes driving genetic diseases. There could be a few more. But the humbling aspect for all of us who are trying to work, all of us physicians, scientists, and biologists who are trying to work on rare diseases, is that really, I think we would be generous with ourselves if we say that we have treatments for about 500 of them. So as you can appreciate, there's a huge mismatch in the numbers between the size of the problem, many thousands of genes causing diseases, millions of people around the world affected by this, and yet we haven't really made a lot of progress in building treatments. And so, in some ways, the reason that I mentioned that the Human Genome Project has provided a huge advantage in the study of rare genetic diseases is because now that we have a map, a reference for all the genes in the human genome, we can easily identify a child or a person with a rare genetic disease because we can find the misspelling in the DNA that's causal to that disease, right? In that one single gene. So because there is a single causal mutation, it is possible to then work on the biology, like what goes wrong inside the circuits of the cells that makes them sick and ultimately causes disease, right? That's how does the DNA mutation translate into something faulty inside our cells that ultimately leads to disease. So this turns out to be a tractable pathway. This is what I've devoted my career to doing, understanding those mechanisms. So as Rachel said in the introduction, cellular homeostasis means how cells are, normally, healthy. And then in the case of mutations, how those mechanisms of homeostasis are disrupted, what goes wrong in the circuits of the cells that ultimately leads to disease. So that's what I do. This is what my lab does. But it turns out that to date, and this is true for me and my lab as well as many other scientists around the world who are working very hard on the problem, the issue is that we are all doing what I call hand-to-hand combat with one gene and one mechanism at a time. And so it turns out that that takes a really long time. So it can take a decade, for example, to go from understanding a gene that causes a rare genetic disease to ultimately understanding what goes wrong inside cells as a consequence of this mutation so that you can ultimately develop a way to fix it. And unfortunately, this is very inefficient and time-consuming, as you can imagine. And as I also tell my team, the patients are waiting. And so we cannot possibly continue on this path because it would take many lifetimes, many scientists' lifetimes before we can actually solve all 8,000 rare genetic diseases. So what can we do? Well, it turns out that we have a lot of technologies. So I'm sure that in the last few days, it must have come up or in your other readings or discussions. We have technologies like CRISPR, for example, that allow us to do things at a scale that was not possible before. Being able to perturb every gene in the human genome at scale in cells using CRISPR technology, for example. So we have tools that we can now deploy. And the hypothesis or thesis for this new initiative that I'm building is really around the idea that maybe we don't have to do hand-to-hand combat with each of these diseases. Maybe we can use our scalable tools to try to figure out how many of these diseases have something in common. And once we understand that common pathway, what I call a nodal biological mechanism, then maybe we can develop a therapy that will in turn help many of these diseases at once, rather than doing them one by one. And I will show you an example for why I think that this could be a tractable approach. It's inspired by my own work. And I hope that we can use this as the foundation for building this new initiative to make faster progress. So any of you who may have done any research, or if you Google my name, the first hit will be my TED Talk, and you will recognize this picture from my TED Talk. So this is actually the Nelson family. This is Roscoe Nelson, his wife, and their 6 children. Roscoe Nelson was a surgeon, but he died in 1960 from a mysterious disease. He knew that his kidneys were progressively failing, but he had no idea what the cause of the disease was. And, he had no access to treatment at that time. And several years later, within the next decade or two, 4 of these beautiful 6 children also developed kidney failure, just like their father. And so this became the inspiration for a journey that my team took to understand the gene that causes this rare, in this case, genetic kidney disease in this family and many other families like them, and then go on the journey of trying to understand the mechanism, what goes wrong inside kidney cells, and then we found a way to treat the disease. But it also became an inspiration for nodal biology, which I'm about to share with you. So this is the family history, just to say that as far back as the 1800s, every generation in this family has been affected by this terrible, lethal kidney disease. And yet nobody really knew what the cause was, right? It took the firepower of my colleagues at the Broad Institute, again, using this incredible tool, our knowledge of the human genome, right? Being able to read every letter in the human genome, that we finally were able to identify the mutation. And it turns out to be in a gene called MUC1. This light blue circle is supposed to represent a cell. The even lighter blue circle is the nucleus of the cell, where the DNA lives in each of our cells in our bodies. The fluorescent green is supposed to represent a piece of the DNA where there was really a single misspelled letter in that part of the DNA of the MUC1 gene that caused a whole lot of trouble. And so what we were able to identify is basically that the mutation is leading to a misshapen, squiggly green protein, as shown here. And it turns out that this protein is bound by another molecule called TMED9. The details don't really matter. But on account of the binding between these two things, the red and the green thing, there's an entrapment or an accumulation of this misshapen protein inside kidney cells. And that turns out to be toxic to the cells. It's almost like accumulating a whole pile of, trash inside your cells that you can't get rid of. And over time, the cells, this becomes toxic to the kidney cells and the cells die. And so this is the mechanism that we uncovered. It took the better part of 5 to 6 years to figure this out, doing hand-to-hand combat in the lab, as I described before. And once we had this understanding, though, we could actually now look for a way to fix this problem. So we turned our attention to the question, can we find a way to get rid of the misshapen toxic protein from these kidney cells? And after some more work, we did, in fact, find a way to do this. So all of this accumulation that was toxic, we were now able to find a treatment, a compound that could direct all of this toxic waste, if you will, into the cell's trash disposal system, which is called the lysosome. So we found the compound that was able to direct all of these misshapen proteins into the cell's lysosome to finally get rid of it. This is now a treatment for this kidney disease, at least what we were able to uncover in the lab, right? Again, several years to do this work. This is a mouse that has the same disease as the Nelson family and all these other patients. There are millions of patients around the world with this disease, we estimate. But this is what happens to the mouse kidney. The green is the toxic protein. If you give this compound to the mice for 1 week, this possible drug to the mice for 1 week, this is what happens to their kidneys. You can get rid of a lot of this toxic protein. So we were obviously very excited about this because it was a discovery that could help us help these patients, ultimately. But what was most catalytic about this, and this is where this nodal biology concept comes around. I don't really have time to tell you how we figured this out, but we basically figured out that this entrapment of a misshapen protein inside cells is not unique to this kidney disease, and it's not unique to kidney cells. We found that the exact same entrapment mechanism is true in the eye, causing blindness. It's true in the brain, causing some forms of Alzheimer's disease, for example. And we continue to do work and identify more and more organs in which this kind of mechanism causes what we call toxic proteinopathies. And it turns out that there are literally dozens and dozens of additional diseases that can benefit from the same therapeutic approach because they are all caused by the same nodal biology, the entrapment of misshapen proteins inside the cells, right? And so this was the great insight, if you will, that led us to an understanding that we no longer necessarily have to do hand-to-hand combat if we go and systematically find the nodal biological pathways that can serve many, many diseases at once, right? So that's the concept, that's the realization that came from this. And just to show you that this is really true, this is actually a mouse eye that has blindness, the same as humans. It's a disease called retinitis pigmentosa. And this is what happens when you treat the mice with these compounds. So this is now not the kidney, but the eye of a mouse with a different disease that causes blindness, but has the same underlying nodal biological mechanism. And as you can see, we can get rid of a lot of this green toxic protein from the eyes of these mice. So with this realization in mind, we realized that we can now go after many, many more diseases. In fact, we estimate that there are probably more than 50 diseases, rare genetic diseases affecting different organs that can benefit from this approach, this nodal biology. And of course, then what we wanted to know is, well, wait a minute, can this approach be applied across all rare genetic diseases? Can we go and find more of these clusters of diseases that could be served by the same mechanism? And why would we want to do that? Because the patients are waiting. This is just the Nelson family at their annual reunion. And you can just see, I can tell you based on the genetics of this disease that 50% of the people in this picture are going to have kidney failure or have kidney failure. And so you can see how dire it becomes when you take it down to the level of the patients waiting for treatments, that we all have to work harder and get clever about the ways that we can develop treatments. And so the fundamental question for what this initiative is all about is, how can we scale our efforts to more rapidly address as many of these rare genetic diseases as possible? Can we tackle all of them? Could that be possible in some way? And so this is the mission of this new initiative called Ladders to Cures. At the present time, it's an initiative that comprises all the scientists, hospitals, and universities in the Boston area, but we hope to go national and ultimately international with this effort, because it would truly take the efforts of literally hundreds of scientists around the world, maybe even thousands of scientists around the world, until we can finally have treatments for all patients with rare genetic diseases. But this is the mission, to catalyze progress as much as possible. I did want to say that we already gained national attention with this effort. We had an inaugural symposium that was in Boston. However, there was a virtual aspect as well, and we had 1,030 participants from across the United States interested in learning how we can achieve escape velocity, as I call it, for rare genetic diseases. And it has definitely sparked new ideas and collaborations, which we continue to work on. This just happened this past summer, a few months ago, and we're now building on that momentum to continue our efforts in this space. And so the final slides that I want to show you, and I don't need to dwell on this so much, but as a subset of our efforts under the rare genetic disease umbrella that we call Ladders to Cures, one of these efforts is a project that we call Project Radical. And this really came from this symposium. I wanted to give you an example of a really ambitious, big idea that came from this 1,030 people coming together and putting their heads together to figure out how we can make progress. And so we decided that there is a way to actually simultaneously interrogate all 8,000 genes causing rare genetic diseases. And we hope to actually introduce these mutations into human cells and then study them with different profiling technologies that we have available to find those shared nodal biological mechanisms. So we're trying to take 8,000 things and put them into bins of 50 here, 200 there, 500 there. What are the ones that are more like each other, so that they presumably share this nodal biology. Once we have these, then we can identify the specific molecular nodes and then build therapies specifically for these nodes that will now serve all of the diseases that were binned together. That's the idea of this concept, this project. It's never been done before. That's why we call it RADICAL, although it stands for Rare Disease Cures Accelerator. But we want to take this approach and take a stab at doing this. It's as ambitious as a human genome project type of thing. It will require a lot of scientists working together, but we're already starting to put the building blocks together, and we hope to continue to make progress in this direction. So this is what Project Radical is about. This is a little bit more of the workflow on this project. I don't need to go into the details. There's a lot of, detailed scientific methods. I'm happy to if you have questions. But the idea is that we actually have some very concrete plans and ideas about how we might go about targeting 8,000 rare diseases at once to try to identify targets that can ultimately be targeted for therapy. And the important thing that I wish to share with you is that we are going to take all of the information that we will generate from this project and put it into the public domain. This will be shared freely and openly with everyone in the world. Patients, physicians, scientists, anyone on the planet who wants to mine the data set that we will generate will have access to this information. So this is the project that we're building to try to accelerate progress in this space. So that's the comments that I had for you today. I think that it will be more productive if I try to answer your questions or provide any additional details or explanations. And I did want to mention that if you want more information, you can also visit our website and find more information there. So I thank you for your attention, and I'm delighted to take any questions you may have.
Thank you for that presentation. I am really intrigued by the Ladders to Cures project because it's the kind of initiative that is relevant across so many different areas, genetic disease, family disease, um, when it comes to issues of access to care or access to the ability to be screened and figure things out. It's so important. I'm going to wait to see if there are any Zoom hands, but until then, I wanted to ask you a question that I put to one of the researchers yesterday, Stefano Ravella of Children's Hospital of Pennsylvania, and I asked him, what was it about studying disorders of the blood that seems to be yielding such incredible, sort of exponentially innovative advances in this particular moment in time. So I'll ask you the same thing about kidney disease research. Is there something about the organ itself that's making it a little bit more accessible for researchers, or how would you quantify that?
That's a good question. I would say that this is not the case, and kidney disease is actually severely underserved, both in this country as well as around the world. So I like to pick hard problems. So I picked the hard problem. It was the science that led me to this, to my interest in kidney disease. But now, as you can see, we're studying brain disease and, eye disease and other diseases. But the blood is particularly accessible. And so it's wonderful that we're making so much progress in areas like sickle cell disease, for example, which affects so many parts of the world, and many other blood disorders. The kidney actually is not as easily amenable to a lot of these therapies. For example, no one to this date really knows how to do gene therapy in the kidney, which is obviously some of the efforts that are happening using CRISPR technology to essentially cure sickle cell disease. That's wonderful, and that's definitely something that we're able to do because the blood is so much more easily accessible. So I would say that this is one of the barriers for the kidney, and something that we have to continue to work on. But this is true for many other organs as well. And so, I don't want to make it seem as though kidneys are the only hard one to get to, but, lungs are hard, for example. There's a lot of other organs, like the pancreas is very difficult to target. Even the brain, we have some approaches, but it's not so straightforward. And there's a lot of ongoing efforts in that as well. So there are many, many devastating diseases that affect many different organs. We have made the most progress in the blood because of its accessibility. And so I think that one of the big pushes for the Ladders to Cures initiative is really to try to to solve the hard problems, get to those other places, or tackle those other diseases. I also want to make a comment here, and maybe it will come up in the discussion later. Of course, we are very much interested in these new technologies that can allow us to cure the disease forever using gene editing or gene therapies and things like that. But I think we all also recognize that not only are there scientific and biological barriers to fixing these problems in certain places, like the kidney, but there are also access issues, looking at a societal problem, and also just the burden, the cost of these therapies is really enormous at this time. And it's not likely to drop anytime in the future because their production is just so expensive, the process by which we make these therapies. And so I've been saying this to whoever will listen because I think it's really important, and also for accessibility to other parts of the world. Small molecules, the pills that we take, which they seem old-fashioned now, are, however, some of our best therapies. And they're the ones that we can manufacture quite a bit more cheaply, and we can make them available to everyone in the world, hopefully. So I do think that there's still very much an interest in doing that. It's an unanticipated and unfortunate consequence of new legislation that was passed in this country a couple of years ago, that there's been a little bit of a disincentive for small molecule therapies, as we call them, pills, essentially. And I disagree with that. I think that we should continue to invest in that because it will help. And for example, even for sickle cell disease, there are pills that can be enormously helpful, new medicines that have been developed. And so we don't necessarily need to deploy, gene editing for everybody, right? And it would seem to me that there are many parts of the world that would, enormously benefit from the relief that comes from, the new pill approaches to sickle cell. For example, I don't mean to pick on one particular disease, but it is one where issues of access are just so profound that it's worth thinking about. So yeah, I want to say, as we're excited about the new technologies, we also should think about the older technologies that are still very efficacious and interesting, and potentially can democratize access a little bit more easily as we move forward. So that's certainly at the center of all of our efforts in our Ladders to Cures initiative.
I want to restate something you just said to help journalists understand, researchers like you said, I like to pick the hard problems. And that is so important, particularly in this realm, because when you think of the numbers of conditions that are still out there to be found, people like Anna are so important in that process. Let's go to Karen Weintraub of USA Today.
Hi, thanks so much for joining us. I have sort of one broad question and one obscure one. The broad one is, where does your treatment stand? What's the progress of that? And the obscure one is, does it help? Could it help Sonia Valeb in your own facility?
Oh, Sonia. Yeah.
And I can tell a little bit, or you can tell a little bit about her story.
No, you should tell Sonia's story, but yeah, I can answer the first question. So we have, of course, ongoing efforts to try to transition this new therapeutic strategy into clinical trials. And all I can say is that it's kind of in the works at the moment. Nothing is in the public domain yet, but there is efforts in stealth mode in the Cambridge ecosystem, if you will. So there is an effort to bring this to clinical trials, and we're very excited about that, of course. And the patients are excited, and the investigators are excited. I think it's going to be wonderful to continue to push this forward, and it's going to be like a small molecule or pill approach to this disease. Sonia, I'll let you tell the story. It's incredibly compelling, and I'll let you tell that story.
Sonia is somebody who now works in the same building, I guess, as Anna.
Yes, we're friends.
Her mother, when Sonia was in her early 20s, I guess her mother was diagnosed with a rapidly progressing neurological condition. She died within a year of diagnosis, and it turned out that Sonia had the same genetic gene rare disease, and was going to head in the same direction. Sonia and her husband, newly married, both changed their whole career trajectories, got PhDs at Harvard. They were not dummies, obviously, and they both worked at Broad. They now are still at Broad working. They now both have their PhDs, and they're working toward a cure for Sonia. But it sounded like what you just came up with might be a treatment for her.
Yeah, so Sonia and Eric. So Sonia was a lawyer and Eric was an engineer, and they both retooled to become scientists like me, and they're now faculty at Harvard Medical School. So they are my colleagues and wonderful, wonderful people. And they're sort of in a race against time, if you will, to find a treatment for Sonia, and many patients like her, obviously, right? And they're incredible advocates. So this therapeutic approach that we have may possibly be helpful for Sonia's disease, but the approach that they're taking is more definitive, actually, because they're really trying to kind of reduce the bad protein in the brain in a way that this approach may not be as effective. So while we keep it in our back pocket, I am very much in support of their efforts in using another kind of technology. Antisense oligonucleotides is what they have settled on for their approach, and they're making excellent progress. But it's a wonderful example of another way that one can pursue hard problems, like Rachel was talking about, which is the patient scientist, right? That's kind of an incredible, Sonia is a patient scientist. It's a really remarkable story. So thank you for bringing that up.
That's actually another good story idea for journalists to explore and find out if there are any patient scientists in their countries who are doing this work for very specific reasons. Let's go to Nadima from GH1 TV in Ghana.
Right. Hi, everyone. The whole process sounds so exciting. Gene testing sounds very fascinating to me. I'm just wondering whether you get more persons with rare diseases presenting themselves up for testing, or the research is mainly on animals, like in your presentation, the example you gave us. And then recently, I engaged a researcher, and the first question he asked me was, why rare disease? Why are you focusing on rare disease when we have a number of conditions as well? So it's like there's not that much interest when it comes to more scientists or researchers going into research efforts towards rare diseases. What do you think can be done? And do you think there's also a collaborative efforts like globally between scientists or researchers to get the work done, or basically the work done currently is done in isolation? Yeah, thank you.
Oh, thank you. This is a really wonderful question, and it gives me an opportunity to talk about the great value of collaboration in rare diseases. But let me first tackle the first question. So rare diseases are by definition individually rare, but as we discussed, and I know you probably have discussed over the last couple of days, once you look at them collectively, they're actually quite a significant problem. You know, they affect 10% of the population. I think we haven't captured all the patients with rare diseases globally, because the current numbers stand at around 300 to 400 million people. But given the fact that there's 30 million people in the US alone, and the global population is 8 billion people, I suspect that the numbers are a vast underestimate. I think that there's probably far more than 300 to 400 million people around the world. And of course, there are places where even the diagnosis is inaccessible to patients, their families, and that's tragic and something that we have to fix. But the idea is that, of course, this is a much bigger problem than each of the diseases individually. And this is actually the impetus for trying to find ways not to do hand-to-hand combat, as I described earlier, but trying to take a more kind of global approach to rare diseases that might yield faster results. So that's just one thing to say about the size of the problem. The second thing that I want to say, and again, if anyone hears my TED Talk, you'll see me say that again, it's a theme. They should be a research and scientific priority for all of us. And I will tell you why. Each of these rare genetic diseases represents nature teaching us about an important biological mechanism that is important for human cells to function properly, right? And so every time there's a defect in the DNA that causes a disease or some kind of faulty circuit inside the cell, this is a way for nature to teach us that we should pay attention to that pathway or that mechanism inside human cells. It turns out that all the common diseases that we worry about, which is what the researcher was telling you, Nadima, heart disease, cancer, diabetes. Of course, we have so many, millions of people around the planet with these problems. All of these diseases are caused by faulty circuits inside cells that have already been nominated as being important by these rare genetic diseases. And so I think that the advantage to researchers comes from being able to move from the rare to the common and take those lessons from the rare diseases and apply them to more common diseases. And to try to make this a little bit more concrete, I would say, if there are 3 rare genetic diseases, I'm just giving you an example, that nominate 3 specific pathways that are important for these, let's say brain diseases, for example. It turns out that these pathways that they nominate, these 3 pathways, might be extremely important to target for Alzheimer's disease, for example, which, in most cases is not a genetic disease, except for rare occasions. But what the rare forms of Alzheimer's are teaching us could be extremely important for those common versions of Alzheimer's disease that so many of us know, in our relatives and family and friends that they suffer. So the drugs that we will develop for those 3 rare pathways, if you will, can be used in combination to treat patients with the more common version of that disease. So you can immediately see how we can take lessons from one space and apply them to the other. And so all of these efforts are very well worth it. And so not only for the sake of rare diseases alone, but for what it will teach us about human biology and how it's disrupted in disease, this is an area that is very well worth the effort. So I definitely think that that's an argument to make to researchers who say, why waste your time on rare diseases? It's not a waste of time at all. It's actually extremely valuable, both for rare as well as for common disease patients.
Let's hear from Santiago Nguyen from Vietnam.
Hello, doctor. Because I'm concerned about the problem of sharing patient data about rare diseases across the globe, so that you and your colleagues may shorten the time to drug development, or maybe something else. And how about the data accuracy concern? Do you think it's accurate? Yes, it's all.
Thank you. Yeah, it's an excellent question. And actually, I didn't fully even address the previous question, which is a good segue into this one as well. So the international collaboration piece is really important for rare diseases, because we need to go where the patients are and try to gather the data from the patients. So for the example of the rare diseases that my group works on, the ones that I just described in my talk, when we started to do the work in cells and in mice, the first thing that I did is that I actually contacted the doctors who take care of these patients, first here in the United States. And I said, we need to build a registry. That means a data repository, exactly to your point, Santiago, basically to gather information about these patients and understand how their disease progresses. Basically, we need to use patients as our teachers, because how else will we know exactly how to deploy the therapies in order to help them if we don't learn about the progression of the disease? How fast or how slow? Does it affect children or adults? Are there any other specific considerations that we have to take into account as we think about designing clinical trials? So we built this registry, and then this registry now contains thousands of patients from all over the world. How do we make sure that it's safe? So there's specific software that is available. And in fact, this is part of our Ladders to Cures initiative, to create these spaces that allow data to be stored safely, right? And it is the data are saved in a way that what we call federated. I don't know if you have heard this before from other spaces, but federated data means that the people from, let's say, the people who are in Vietnam or in the United States or in Australia or in Germany, they can contribute the data from their patients into this common repository, but they also have complete access to their own data because the system is federated. So everybody owns the data from their own cohorts. But there are parts of the data set that can be shared in an anonymized way, of course, across the entire group of investigators. So let's say I am sitting in Boston and I want to know about, how many teenagers have this disease, right? I say I want to find out how many people between the ages of 13 and 19 have this disease. I can go on this federated database and I can enter this query, and it will give me the numbers in the United States, in Germany, in Vietnam, in Australia. I don't know who these patients are, and I don't have access to any of their personal information, but I can get an answer to my question, right? That there's 2,000 patients in the registry with that. And then I can ask additional questions like, how severe is their disease? Or when were they initially diagnosed? Things like that, right? And so the power comes from being able to bring the data together. It absolutely requires international collaboration, which is, what we just discussed earlier as well. But the safety and the safety of the data, and the fact that the data have to be, anonymized in a way that it doesn't in any way compromise the patient's privacy, is something that we're very focused on and we continue to build on. So this is a very important topic.
I'm going to ask Julia Metro of Mother Jones to jump in and make her comment.
Yes, hello. So I have a condition called vasculitis, and I just want to comment on how a rare disease thing can suddenly become a general population issue. So vasculitis is a series of 20 disorders, might have a genetic component because, as we know, it's more prevalent in certain ethnic groups. I wouldn't say we all know. I know this. I spent way too much time reading up on it. But because it's a blood vessel condition, especially early on in COVID, when people were trying to guess the etymology or like how it's working and all of that, I know my specialists have been very much on call. But also understanding how blood vessels work and all that from rare disease research was able to be implemented, and also like knowledge about, okay, if you've experienced very acute inflammation in your blood vessels, maybe prednisone is good to use, just because to stop any organ damage, but it's really something that was like applicable for something that's rare to the general public as well.
Absolutely. I greatly appreciate your comment and I endorse it wholeheartedly. And exactly also what I said in my TED talk, that was the key message, that, you know, the lessons from rare diseases are really applicable to all. And it will help all of us develop precision therapies for all human diseases ultimately. So I strongly believe that. And there are many such examples. Thank you for your comment.
Anna, we're going to have a session on Friday on clinical trials, access and equity, and researcher from Duke University is going to talk about some of their efforts in that regard. And I think you would be really doing us a huge favor if you would talk about, excuse me, how the Broad Institute and you create and fashion, or what is your strategy for ensuring diversity?
Yeah, this is a really key topic, and it sort of ties into the discussion we were just having also based on the question from Santiago as well. I think this is a key point for rare diseases, but for all diseases and all clinical trials, really. You know, the idea of having representative groups from that represent the human diversity, if you will, you know, from different ethnic backgrounds. You know, we can't be doing trials in only men or women, you know, like there has to be, or only adults and not children. That's a huge issue actually for the pediatric colleagues, that this is often the case, that, you know, we don't have enough information about diseases in children. And yet in the case of rare diseases, like upwards of 80% of these diseases affect children. So it behooves us to include them, of course, in our clinical trials. So part of the reason why we're building ladders to cures is actually one of our mandates that I didn't emphasize as much in my introductory remarks, is we actually are going through this process of basically making sure that our patients are our partners in the process. And this is a way to democratize the access to information as well as access to future clinical trials. So if the patients are there as partners in the process from the beginning, from the discovery, all the way to understanding mechanisms, developing therapeutic strategies, and ultimately going to clinical trials, this accomplishes several things. First of all, patients are our best teachers, because we physicians may think we understand disease, but we really don't unless we actually ask the patients and we sort of, you know, study closely with the patients about the progression of the diseases. So patients can be our teachers. It also helps patients understand just how difficult and lengthy the process of research really is. You know, most of us develop, you know, an understanding of possible treatments over, you know, the course of many years or decades, right? And so I think it's important for patients to also be witnesses to that long and difficult process of discovery. I think it's important. It is also critical from the process of democratizing access to what we do, not only to put this on open portals, the data that we generate, but also that, you know, patients can then use social media, for example, to inform other patients. You know, this is a mechanism we have used at the Broad for some of our projects. For example, if you look into the Count Me In project, if you Google that, it's presently used for rare cancers, but we hope to advance it across all rare diseases as part of our Ladders to Cures initiative. This, again, is the idea that patients are not just there to teach us about the disease, but they become the best advocates to other patients, you know, and so that sort of creates this open access. And social media, I mean, there are many downsides to social media, but there's an upside too, that patients can find others and inform them about the progress in research. And ultimately, patients can also inform other patients about clinical trials and the availability of clinical trials. And I think many of us know that there are certain, populations of patients who may not have traditionally had access to this information. And I think now this is kind of a new way through social media that we can make sure that we can, have everyone have access to the information and the possible eligibility for clinical trials. So we have a huge mountain to climb, just to be fair, we are not there. But I think we can use technology for good, in this case, to our advantage, to do something good, which is to really, as much as possible, democratize access to the research. And I want to make one more comment, which I think you will find interesting. In our efforts to make patients our partners, this is a project that is currently running in the United States and Canada, but it has the potential to run internationally. There's something at the Broad we call the Rare Genomes Project. You can also Google that, Rare Genomes Project, Broad. And what that does is, if you have a computer and a mailbox, you can participate in our research, because that's all you need. You don't need to be close to a major medical center. You certainly don't need to be close to the Broad. But the way that it works is people sign up on the website, then they are consented over the phone for participation in the research if they're eligible. And then we can send them in the mail a kit, which they can use to either provide a saliva sample or a blood sample if they go to a local lab where someone can draw their lab. And they can send it back to the Broad for sequencing for free, so that we can diagnose rare genetic mutations. And then once the diagnosis is done, Ladders to Cures is this new initiative that will now work on what happens next, developing an understanding of the mechanisms so that we can ultimately develop treatments. But this is one example, the Rare Genomes Project or the Count Me In initiative, one example where, again, if you have a computer and a mailbox, this sort of democratizes the process of participating in our research, which I think is important.
We have a question from Chong Sha Chan of Malay Online in Malaysia.
Hi, Dr. Anna. This question is about how about patients who are at the infant or toddler age? Do you find it challenging as they couldn't express their feeling after the therapy?
Mm-hmm. Yeah, this is an interesting question. So, of course, the parents are the ones who consent for children to participate in research or in trials, and that's complex, of course, because the parent has to basically advocate for their own child and represents the interests of their child. This is why infants and toddlers are generally not the first place that we go for enrolling patients into clinical trials, because it's very difficult to understand their responses sometimes to treatments or the side effects of those treatments. However, there are approved therapies that have been given to toddlers, for example. So there's gene therapy that has been injected into the spine of patients with a devastating disease that makes them unable to walk. And these toddlers are now able to walk on account of that therapy. And so that's a way that you can demonstrably measure the benefit. A baby that is losing the ability to move and now can walk, that's a huge gain, right? Some of the other things are a little more subtle, especially neurological abnormalities, neurodevelopmental defects in babies. That's really hard, right, to kind of figure out. Many of these disorders in infants and toddlers manifest as seizures. And so this is a very devastating effect of circuits in the brain that are not working. The babies have seizures, and the parents know this very well because it's very, very difficult to manage those symptoms, the seizures. So in some cases, when medications are working, one of the ways that we measure progress is that the babies have less seizures or hopefully no seizures. And so I'm just giving you some examples to say that there are ways to measure progress, even in babies and toddlers. Of course, it's difficult, and most trials don't begin at those ages. But in some cases, they must, if we're going to help these very small patients derive the benefit that they can. Because you see, sometimes the genetic disease advances so fast in those early years that no matter how good the treatment is later, it's past the opportunity to really fix something, in the brain, for example. And so sometimes you have to be able to deploy the treatments at those very early years of life. There's a lot of complexity around this, of course, and all kinds of issues, ethical, legal, clinical, scientific, there are many aspects to this. But, we have to take it on, again, take on the hard problems and help as many of our fellow human beings as possible. So that's the goal of this initiative.
As we start to wind down, make sure if there are any other questions, put up those Zoom hands. But I wanted to jump in and talk about the issue of disease burden, sort of a geographical look at this. And yesterday, Dr. Ravella showed us a map, a global map. And of course, for sickle cell, it's, the map was sort of nicely colored, a few dots here in North America and a few in South America. But then there were these huge red and orange balloons on the African continent and Southeast Asia and whatever. And so I'm wondering if there's a parallel when it comes to kidney disease.
Yeah, there's actually a really wonderful parallel in kidney disease. It so happens specifically for kidney disease. Some of you may know that the reason that evolution allowed the sickle cell trait to exist in humans and be prevalent in that part of Africa is because it confers an advantage to survival from malaria. I don't know how many of you know this, but the sickle trait would, make people more resistant to malaria, but then unfortunately it causes sickle cell disease. So these are the trade-offs that happen sometimes as evolution occurs in all humans over time. So a similar question of an advantage for one pathogen that confers disease on the other side is actually in kidney disease with a trait that actually evolved in West Africa, specifically in the Yoruba population in Nigeria. And this is called the ApoL1 trait. And so if you have this particular mutation, you are more resistant to trypanosomal infections, which are prevalent in that part of the world. However, this same mutation confers risk or susceptibility to kidney disease. And so the reason for a large proportion of people in Africa, as well as in the United States and other parts of the world where people, who have an ethnic background from West Africa come from, is because of this, again, this coevolution of this trait, right? So there's an absolute parallel to sickle cell disease in kidney disease, and ApoL1 is a great example of an area where we can actually have a huge impact. Certainly in the African American population here in the United States in terms of treatments for kidney disease, but also in populations in Africa in particular, where kidney disease is highly prevalent. And so first, the discovery of this was a major discovery. Again, the genetics helped a great deal. And it happened here by one of my colleagues here at Harvard, was the discoverer of this. And now there's several efforts to develop treatments. They're in clinical trials at the moment, but it's extremely hopeful that one of these is going to work. And that can help people, hopefully both in the United States as well as all over the world where there's kidney disease caused by this particular mutation. So
can you spell that for me? Because I want to look that up after.
Oh, of course, yeah. It's ApoL1. It's a very interesting example, similar to sickle cell disease, but it was more recently discovered, so we know a little less about it. But the progress has been great, and so I think that this is it was a perfect question because it gave me the opportunity to tell you about this interesting finding.
Let's take one more, and that's from Rupsha Chakraborty of the Indian Express in India.
Hi, I just have one single question. So in the past years, we have come a long way in developing medicines for rare disease. but still now many of these medicines are unaffordable. So will this therapy will be more cost effective compared to other contemporary treatments that are currently available?
Yeah, this is this is a big problem. I can't profess to have an answer, to this. The affordability of treatments is something that we talk about and worry about a lot. It a little bit comes back to the comments I made earlier in our session. So a lot of these genetic therapies, like gene therapy, gene editing therapies, are gonna be unaffordable, basically. It's going to be very, very difficult to be able to make them widely accessible, at least based on current resources, and how expensive it is to produce them. But there are other treatments, like, in the form of pills, for example, that are more affordable and that we can make more widely available. So I think that there are ways in which we can advocate for more of these treatments to become available in other parts of the world. And I think, I can't speak for them. Obviously, I'm an academic, but, drug companies should think of this as a moral imperative to try to find ways to make therapies accessible to countries and parts of the world where there's less access to, or that there's less wealth that can support, these medicines. And so, selling them at cost in certain parts of the world. We saw this happen, in the case of COVID vaccines. There were examples where this happened. So I think that we can definitely pursue those approaches. Again, I'm not, in charge of any of those decisions, and I certainly, I'm not in charge of the thinking of on the part of pharmaceutical companies. And I understand that they also have, they're a business and they have to make a profit. I get that. I'm not being, Pollyanna-ish about the realities of how the market works. But at the same time, I think that there is ways in which we could try to make more of these treatments accessible. And this is the reason why I think disincentivizing the development of pills for the benefit of, for example, biologics, as we call them, or genetic therapies, is not necessarily a clever idea, because those are the most inexpensive treatments to make. And so I think that we should continue our efforts in that space as well.
I said that was going to be the last question, but our journalist Taiwo Adebulu from the Cable in Nigeria has forced my hand. He says he is a member of the Yoruba tribe, and he wants to know more about the mutation. So can you briefly, Taiwo, tell her how you want to go about doing a story?
Yes, because I find it something quite new that I have not heard about before. But I knew it's quite true that kidney diseases are quite prevalent in the southwestern part of Nigeria, where the Yoruba tribe is. So I think doing a story about this is going to be quite expository. So I don't know the kind of resource materials that Anna has on that particular discovery. So I would like to have that and go further. So I don't know if I could get maybe your email or something that could make me ask more questions and all that.
You are welcome to email me, but I will do you one better. There is a Nigerian colleague of mine who investigates this disease. His name is Opeyemi Olabisi. He's at Duke University. Opeyemi Olabisi. I'm putting it in the chat. And I'm happy to connect you to him so you can do a story with him, because what is more perfect than a Nigerian American investigator doing work on this specific disease? I mean, it's perfect.
So what I will do, Taiwo, is connect you to Anna after our day ends today. I'll do an email and connect the two of you.
It will be my pleasure. Yeah.
Listen, Anna, this has been an extraordinary experience for me, because as I told one of our researchers yesterday, when someone like you can come in front of us and communicate these issues in ways that we can grasp, we can actually visualize and see the importance of these topics, it is such an incredible privilege. So I want to take this opportunity to formally thank Dr. Anna Greka of the Broad Institute and so many other amazing research institutes for coming to join us today at the Covering Rare Diseases Fellowship. Thank you.
Thank you so much. It was a great pleasure and an honor to be with you. And I wish all of you the best of luck. And please do cover rare diseases in your stories. It's a very, very important topic. So thank you for the opportunity.
Great. I will be in touch with you. Thank you so much.